Today we'll start with the next topic, as we all as teachers as asked by you all. So we'll go to the next. How can we better? There's such as we always discuss. It's a simple discussion about how can we conduct classes in while teaching these principles of inheritance and variation. Regarding that, today's session we are going to discuss, teachers. So this lesson, half of the lesson are already known lesson to the children. They would come to class with already a formed idea. Their idea will be there about like whatever the experiments they have studied about the Mendel's experiment. The other half of the lesson will be completely very new to them, unknown to them. So it's this lesson.
Sir, your voice is breaking, sir.
Grasp it. Is it so, ma'am? Oh, sorry. Am I audible, ma'am, now?
Voice is breaking, sir.
I don't know why today that happens. Let me check. Am I audible, teachers, now?
Yes, sir.
Yes, thank you, ma'am. Thank you. Sorry for the inconvenience. If in between anything such things happens, please let me know. I will change over the network. So this lesson mainly the basic things and all. I think student themselves will be able to understand. Mostly we don't have much work in the first part of the lesson, like Mendelian inheritance. It's just like they will learn it in a very quick manner. Of course, we need to teach them that what is that test cross and all. They need to understand very well. Otherwise, some definitions which commonly they have studied in the lower class, that we will be recalling. Mostly with that first half of the lesson or first part of the lesson is over. The second part actually starts with the non-Mendelian inheritance. What are the deviations are there? From here onwards, student has to be taught in a very detailed manner, as we all always doing. Here, students will have a lot of confusions, lot of doubts, and lot of misunderstanding. Also, sometimes it is possible because they are not into that. They already have learned certain rules and regulation in the 10th standard. Usually, we won't introduce the deviations in the 10th standard. Generally, we don't introduce. Why? Because it will further complicate the teaching. Mostly we don't introduce, but if we introduce also, there is no wrong. If at all we are handling 10th and all we are handling, there is possibility that we can even say there are some deviations will be there in the 10th standard itself. We need to throw the light onto them so that when they come to 12th standard, they will be able to cope up with the syllabus in a very easy manner. So this incomplete dominance, here what is the issue they will have? Generally here they will come to many times they will say blending of character happens. The pink color is the outcome of white and red color contribution. Both the alleles are contributing. That is the reason why the pink color is there. So this, however, we say that there is a generalized idea. They will come to a conclusion that definitely pink color is outcome of red and mixture of red and white allele. Both the alleles express red allele for red color and white will express for white color polypeptide. As a result, both will mix together to form a pink color. That idea, they will generalize very well. So in repeated time, by conducting continuous tests, we need to make them understand it is not equal to mixing of red paint and white paint to make a pink paint. It is not like mixing of a hot water and a cold water to make a warm water. It is not, it should not be equated. It will not resemble either of the parent. It's completely, of course, in the later phases, the sentence are given in the book, but still students will be having always confusion when they are learning for the first time about this particular incomplete dominance. In that incomplete dominance, and again we have to make them what is the possibility. The three possibility. The doubt happens because children do not know how this phenotypic is expressed. They know what is phenotypic, what is a genotype, they know what is an allele, they know all these things they know. But generally, this problem arises when they don't know how this allele contribute for the phenotypic expression. That area we need to give little high weightage. We need to make the children to understand that how these characters are expressed, why our skin colors are in a different shades. At the time, the shade is because of the pigment. The pigment is coded by the gene. So this the student should be able to sequence everything, and they must be able to understand how this genotype is being expressed into phenotype. That though they have studied molecular biology, though they are going to study, they will not have that knowledge. So we need to, once again, we have to put them in the mind how it is expression happens. This CBSE, I don't know why they have kept in this order. There are some, if you look into the textbook very well, you may find that this chapter actually requires little knowledge of molecular biology also. The gene expression, the knowledge is required to understand this particular lesson also. But that unit is arranged in such a way that first this lesson is we need to taught, then we have to teach that next lesson, the molecular level. So it is sometime it creates problem that to make the children understand how these characters are expressed. So we need to put our level best to make the children to understand how the genotype is expressed into a particular phenotype. If that they are clear, then definitely they will be able to keep in mind that allele, out of two allele, only one is going to express when in case of pink color. That gives that shade of the shade. They have to use the word that shade. They should not come to, it's not a perfect pink color, that shade they have to use it, and they will be able to comprehend in that way. And phenotypic ratio, genotypic ratio, they are same. How come it is same? And anyway, we need to draw the flow chart, and we need to make them clearly understand how this phenotype and genotype, both the ratios remains the same in case of this incomplete dominance. And next one is, I believe students will take it in a very easy manner. Co-dominance is very easy topic for them. Why? Because in having already studied in 11th standard, they know about that this antigen present on this RBC, all these things. They know how the blood group is actually blood grouping is done. They know very well. Here, all they need to know is simply what is a dominant allele, which one is a recessive allele. That alone they have to know. If they know it, they can easily answer this question very well. Most of even the very mediocre students also able to say this, understand this topic in a very easy manner. What they have to understand very well, what is the contribution of that A allele and B allele, what makes speciality for the person to have A blood group, and why that I, that recessive allele, what is the role played by the I. So they have to clearly say the sugar formation by dominant allele and no sugar formation by the recessive allele, they must know. And next one is that blood grouping. Yes, ma'am. Yes, ma'am. Yes, ma'am.
Easy definition for allele. Allele.
Alternative form of gene, can we say alternative form of gene? We call it as allele. That can be
Sometimes
very
you are telling dominant gene, dominant trait, dominant allele.
Yes. Dominant trait is that phenotypically what we say. Dominant allele is the allele which is going to be expressed. We can say that the allele which is going to be expressed is called dominant allele. Which is not expressed in the presence of a dominant allele are called as recessive allele, we can say. And allele is a
difference.
Yes, ma'am.
Gene and allele difference.
Yes, ma'am. Gene is a unit of inheritance. We can. I hope the students will be in this way. If we put, they will be able to understand. Gene is a unit of inheritance that is going to express into some characters. Some characters are going to be expressed. That characters have an alternative to a chromosomes are always in a homologous pair only. So they have an alternative. It can be similar pair of allele or it can be contrasting pair of allele. Both it can be. So a similar pair of allele, we call it as a homozygous alleles or homozygous gene we can call. Whereas the heterozygous, there will be a contraction. The two allele will not be similar. But here AB blood group, we have to say both the allele which are dissimilar, but they are all both are dominant. That is the reason why
That's
both
not
the
clear,
alleles are
ma'am.
going to express.
How can we
Yes,
define
ma'am.
allele definition?
Yes, ma'am. That's what I am. The alternative form of gene is called as allele. That is a maximum definition we can tell to the children. If any teachers want to add, you can add.
Allelomorph, contrasting alleles are called allelomorph. Same with
Allelomorph, the
different
same only, ma'am.
characters.
Yes. Yes. Thank you.
For example, for allele genes are always expressed in pairs only. For example, for tallness, capital T and capital T. So
Yes.
we can call these as alleles also, or only
Yes, we need to call them allele only. No, the capital T, A or a B, they are all alleles only, allelic forms only of a different genes. They are the alleles of the gene. They are going to express the called as blood group. What is the character controlled by these two both allele together? They are going to code for the blood group. Same way the tall capital T and capital T or capital T small t or small t, all are alleles only. What is their job is they are going to code for their the trait controlled by these alleles are that is height of the plant. What is the allele is there based on that height of the plant would differ. If both are dominant alleles are there, that is capital T, then it is going to code for tall plant. Like that we can say. But kind request, I am using now only. Please let us not put the word letters alone into their mind. It's what happens, they are only into that. They feel like only the letters are the alleles. They are stick on holding to that particular thing only. They make it as a theory. They don't understand into the allele level gene how the alleles are distributed. They don't understand that. So it's a kind request. Better we not to put that letters alone into their mind. Make them understand an allele. It's a dominant allele. We are assigning the letter T for that. It's a recessive allele. We are assigning a letter small t for that. Like that, if we put, student will be able to distinguish between how exactly it will be present in the on the chromosome teachers.
then for example,
So
if
next
when
one, you
some
must
other
know.
letters also, is it okay? Is it necessary to use only capital T, T?
No, ma'am. No,
or
ma'am.
some other
It is governed by a certain rules that we need to use that only. Whatever is at all, whatever already assigned, we need to use those letters only, alphabets only, for to indicating that we cannot on our own. We cannot give certain letters on our own. We cannot give them. We need to give that what is there in the already ruled. In case of Mendelian inheritance, we are going to follow whatever as Mendel has already suggested. That only we are going to. So this codominance, mostly it is easy. I believe the students are able to, but we need to put lot of questions to them, like involving flow chart, where the students are making mistake. If you see, if you are asking mother's blood group is A, father's blood group is O, what will be the blood group of the children? When we are asking, student must be able to understand clearly that the mother's genotype also they have to understand. They must understand. Anyway, father's genotype, no need to understand. It's very clear that father is having both the recessive allele only. That's why he has got O blood group. When it comes to mother's blood group now, because the example wise I'm talking, so they must be able to understand the genotype of the mother's blood group or the one person blood group is very, very important. Based on that only, the blood group chart can be drawn. What is we can predict the blood group of the offspring. The student must be able to understand clearly. Generally, they will generalize. Mother is A blood group means having allele of both the dominant allele. Father is O blood group means the O blood group only is there. So the child is possibly will have only A blood group. Like that, they will generalize. So we need to make them understand by giving more examples. Questions we can ask. We can write on the board. We can ask many crosses. We can IA with A blood group with B blood group, heterozygous A blood group, heterozygous B blood group, homozygous A blood group with the homozygous heterozygous B blood group. Like that, in different methods, different questions we can frame. 2 crosses we can give it to the children, so that children we can test whether or whatever the given situation they are finding the appropriate answer or not. We can try to find it out. That will make their understanding very clear, teachers, that we can do. Next one is the score dominance. How that, as I said, how can that can be drawn into that conclusion. The genotype is very, very important of the parent to come to a conclusion of, only in case of heterozygous. Homozygous, there is no need. Homozygous, it's very mainly when it is a recessive character, there is no need of any genotypic to find out. It's already clear, it's understood. So next one is the multiple allelism. Blood group can, student must clearly understand, blood group can be stated as an example for 3 different situations. One is the complete dominance, next one is codominance, and third one is multiple allelism. All the 3 concepts they must clearly understand. When a A blood group or B blood group only is expressed, there it is obeying the rule, Mendelian law. So we can call it as complete dominance, codominance in case of AB blood group, and multiple allelism, presence of more than 2 allele for a particular character. Here children must be able to clearly understand the multiple allelism cannot be studied on a particular individual alone. Whatever the number of alleles may be, individual is going to have only one pair of allele, that is 2 alleles only is going to be there, not more than 2. So student must clearly understand the multiple allelism can be understood, very well understood, only when we are doing the population studies. In a population only the multiple allelism can be seen, not on a particular individual. The student must be able to clearly understand why only on that population it is understood, not on the individual. The student, then they will have more clarity that only one pair of allele can be present in an individual. There, whatever the doubts previously they were having also, now it will be clarified. Next, another polygenic inheritance. Here also students need to clearly understand how the presence of many genes can code for just one. As I already said, we discussed during the this molecular biology human genome project at the time. So there itself we need to code that sometime the genes work in a group to control a particular character. Best example, our skin color, our height, these are the things that is controlled. Here student will have doubt, why not the pea plant? In a pea plant, the height is not controlled by multiple genes, only a single gene controls the height. They have to understand. But in human, case of human, there are many genes contribute for that. And the shades of color, how it is happening, that A, B, C, all the 3 genes, all the 3 pairs of alleles are there. Again, we need to put a question to the children, how many pair of allele can be there in a person? We need to ask for one gene. Here students will misunderstand and say only one pair of A, either it will be A or B or C, like that they will say. Then we need to make the children to understand, when we were discussing in the AB blood group, only one pair of gene that controls a particular character. But here 3 genes, the A is located somewhere, B is located somewhere, C is located somewhere. All the 3 genes are there. These 3 genes are together going to control the skin color. So they are not that, we should not misunderstand only one. All the 3, one pair will be present in a person's body. That the student must be able to understand. Same way for the height color. Here we can even add little values to the children. The skin color doesn't matter. It's all genetics. That we need to make the children to understand. That value the student must understand. There's many people will feel like height and all, if you see, students will compare. That is actually, especially during adolescent period, that is very common. So we can add that values to the children, how the skin color and how this height and all is being expressed, so that they will learn to appreciate themselves. They will not have difference of opinion about themselves, and also children will give values to the respect to the others. They won't mock at the others. Next, this what is the scale we can show to the children, and we need to tell the children the cosmetics and all, that is not true. How that the colors cannot be changed in that way, it won't work out in that way. Student must understand. what is the extreme dark skin color and extreme fair skin color, and what is the extreme intermediate color, what are all the gene contribution can be required, they need to understand. Then next very this here what will happen, they will confuse with this polygenic inheritance and pleiotropy, they will get confusion. Here only one gene is going to be there, it is going to express for multiple phenotype, whereas previously many genes are going to control for only one genotype. That students sometimes, some students only usually, they will have little confusion in that. We need to make the children, by repetition only, we need to make the children to be understood.
Tell them, sir. Poly means many. Gene.
Yes,
Many gene.
very nice.
Many
Yes.
gene, one character. We can tell them in that way.
Exactly, ma'am. Very nice. Yes, in that way also we can take some clues to make the children to understand. It will differ on individual teachers. Individually, we may follow certain ideas we may follow. So here, teachers again, if you see what is a very biggest demerit we have observed in biology book, if you see, certain examples will be repeated in many chapters. Say for example, this sickle cell anemia here. The next topic it will be there, and we will be studying, we have studied in molecular biology also. So like that, the same example will be repeated in many areas, that will lead to confusion for the student. In what context they are asking, student must be able to understand, then only they can give a correct answer. Otherwise, the question would have been asked from some other context and student will write some other answer. At last, it will cost their marks for that particular question. So here phenylketonuria and sickle cell anemia both can be stated. If at all children are not writing apart from the textbook, even CBSE recommends that if they are writing sickle cell anemia also, we can give mark, not necessarily only phenylketonuria which is there in the textbook, and sickle cell anemia also they can write for example of this pleiotropy. And even students will hear little confusion will be there. We need to make them understand Mendel was only giving weightage to the whether the seed is round or wrinkled. He didn't give important weightage to whether the size of the seed is smaller or bigger. That he failed to understand, or he was not able to give appropriate answer. It's not known, but he didn't understood that incomplete dominance present in the pea plant. Students need to understand without getting confusion with what they have studied before. What they have studied there is starch produced or not produced, and the first one, round seed and wrinkled seed difference, either the starch is produced or not produced. But here we need to say whether the starch is produced more or it is produced less. So this idea student must be it will take definitely a time. Comparatively, we have to teach them. It will take time, but they need to understand the previously what they have studied, the cross between a round seed and a wrinkled seed. It's a competition between whether the starch is produced or not. But here it is mainly focused on how much starch is produced, whether more amount of starch is produced or less amount of starch is produced. In that context, if we look, we will get the answer that is a less amount of this example will fall under incomplete dominance, which Mendel failed to understand, that we need to tell to children. Then there is a paragraph, there is a line also is there about, and they CBSE itself said not to get confusion, that we need to put into the mind of children carefully. Next one is, this is directly book back only, the book concept only, why the Mendel's work was not recognized for more than around 30 years, 35 years. It was not accepted by the Royal Society of London. They didn't accept his work as the work. Only the first and foremost reason is the usage of mathematics. So we need to tell to the children, biologists did not approve using of mathematics. Same way, mathematician will not accept the usage of biology. This interdisciplinary fight was there for the ages, that the student must be able to understand, and that they should try to overcome that. So because of mainly statistical approach, it was given everything. Mainly his work was not at all accepted in the beginning. And the factors, he was not able to prove a raw evidence or a very concrete evidence for this gene what is the factors are there. Students are always writing. They write instead of factors, they write as a genes when they are talking about Mendel's inheritance. We need to repeatedly tell them when they are talking about Mendel's inheritance, they must use the word factors only, not the word genes. That they must know. The next one is this chromosomal theory of inheritance. Sometimes students are complicating as if something they are going to learn, something very marvelous and very this elaborate thing. But it's a very simple thing that they need to understand. Later, Mendel's work was published, republished. Then only the discovery of chromosome has happened. The chromosome, understanding of chromosome gave a very clear idea what is passing. Mendel was able to say the characters are transmitted from one parent to offspring through gametes, yes said. And who is carrying these characters later came to an idea that is because of chromosome. So this chromosome also work in the pair, and the segregation of chromosomes are happening. Because of the segregation of chromosome only, the characters are segregated. This only was discovered by this scientist, but not about the gene. The gene was yet to be discovered, and the student should appreciate step-by-step discovery. Overnight magic cannot happen. First, Mendel gave some idea. When scientists were doing research on that, they found about something called as chromosome. Later, they discovered that gene. So everything will happen in a stepwise manner. The student must be able to appreciate that efforts of the scientist. Then the chromosomal theory of inheritance also again obeys the laws given by the Mendel. It is not mainly focusing on this Morgan's experiment. They say clearly, they said the independent assortment happens in the chromosomal theory of inheritance. Also, the independent assortment happens. How they are segregated during the gamete formation are very well explained. We can show some this and all will require a little amount of coloring. Usually what I do, just I'm sharing with you. If possible, you also can share your opinion. I will take different colored chalk pieces to the class. I'll make a chromosome having the colors of different gene. The different colors will indicate different gene, how they are segregating. When we are seeing that, student will be, or we can give some model example of some characters. It can be plant height of the plant, color of the seed, shape of the seed, shape of the flower. Like 4 or 5 characters we can take. We can draw the genetic gene map we can make on the chromosome, and then we can make the children to understand how they are segregating. Here we need to quickly make the children to recall what happens during meiosis prophase 1. What happens? Children would have forgotten, which they have studied in 11th standard. So we need to recall that recombination concept, how this recombination happens. That and all quickly we need to recall, so that children will have a little idea they will be able to understand. The next one, this book diagram only. For this also we can use different color chalk pieces or color papers we can use to denote a different gene. We can use all this thing, or probability with the help of some polythene bag or something. We can take this balls, different small balls are there that we can take it to the class, and we can make the children to understand it's all matter of probability, how they are segregating. That we can try to make the children understand. This purely based on, it's all completely based on the recombination only. The segregation is completely based on how recombination, what genes are going from the sister to non-sister chromatid. This all completely it's a matter of probability only. So we need to tell them possibility. These are all possibility. How come the genes segregate present on different chromosomes? Then Morgan, we need to hear again, we need to say Mendel's law is once again disproved by Thomas Hunt Morgan by taking example of this Drosophila, which is, we need to compare why Morgan has chosen this particular insect. Why did Mendel choose that Pisum sativum? Both have the similar, almost similar reason. What was the reason? We can make the children to speak. Children will give an idea. Children will say it will easily reproduce. The life cycle is very shorter. That is the reason which we have seen already there in the Mendel's inheritance also. So the students will be able to very quickly recall some of the points. The other points we can add on to that, like growing on the synthetic medium and less breeding time, and many progenies can be produced, and mainly the male and female will show sexual dimorphism. Easily we can identify which one is a male and which one is a female. And there are many, same way in the Mendel's law, there are many visible characters are there in the Pisum sativum. Same way it is there in the this Drosophila also, that we children must understand. And male and female Drosophila, they must be able to distinguish. Which one can we say, teachers? How will we distinguish? Can anyone say? How can we distinguish male and female? Yes, Amuda ma'am wanted to say something. How can we distinguish male and female, teachers, of Drosophila?
Eye color.
Yes, okay. It's based on the size of the body, teachers.
Sir. And Y chromosome will be long. That Y chromosome will
Yeah. No, visibly, ma'am, by seeing at the laboratory. I'm talking about in the laboratory. How can we distinguish between a male and female? Very simple. Male will be always very smaller. In the book also, the diagram would have given. You can see that male is very smaller and female will be very bigger. That is a very easy distinction. That is called sexual dimorphism. But the moment we must be able to identify between, the student must be able to identify which one is a male and which one is a female by looking into the size of the insect. The bigger is female, smaller is the male. Student must definitely know that. And the different phenotype here, what mistake is happening mainly in the book, they have not given clearly. What are all the phenotypes are there? The characters, like in the Mendel's inheritance, they have given a very clear the table, height of the plant, 7 characters they have given very clearly. But for this particular insect, they have not given. They have given here and there somewhere, like this white wing, white eye, yellow body, white eye female, like that somewhere they have given. So please, teachers, before itself, make the children do what are the characters are there. The body of the organism can be yellow or it can be brown. The color of the eye can be either white or it can be red. The wing size can be small, that is miniature, or it's a large. Which one is a wild type? The student must know wild type is nothing but they are talking about dominant character. And what is that opposite to the wild type, ma'am? Can anyone say? So we need the recessive, the wild type only. In the when the first started, everything was a wild genes only. When a wild gene, when the mutation happens, then it leads to recessiveness. So we can call that recessive gene as a mutant gene also. Wild type opposite, we can say mutant. The recessive characters are the mutant. They are all formed because of the mutation only. They are all the outcome of mutation. When the world was formed or when the organism was formed, maybe they all had A and B blood group only or A blood group only. When a mutation happened, that the O blood group has formed. That mutant gene, wild gene has undergone mutation to form the recessive allele. So we call a mutant gene. So children must understand wild opposite is mutant, wild is dominant, and that mutant is a recessive. What are all the characters are there? Before itself, we have to tell to teachers, please, definitely most of you will be facing a little, maybe little challenge while explaining this topic, I believe so. So that we can overcome by explaining, first of all, what is the character. Then we need to explain the chromosome map of it, how the chromosomes are located. 2 different chromosomes we have to draw for the first character and second character. How far they are located on a same chromosome and a pair of chromosome, how close they are located or how far they have located. First, we need to explain these things. Then we need to say what is linkage and recombination. Then student will understand very easily. And also we can tell to children, though we are all different from our parent, we can very clearly see that some of this will say that you are looking like your father, you are looking like your mother. Few characters will be looking in a person like either a mother or mostly like a father or mostly like a mother. Why this happens? Why some characters are exactly looking like that particular mother or father? What is the reason? We can put forward the question. Then we can explain that the genes which are located very close to each other, which are tightly packed, tightly linked, there is a chance of segregation. Their segregation is very poor. They won't easily segregate. As a result, together they will be inherited. Whereas when the genes are located at two different, far away from each other, two different genes controlling two different characters, when they are located in a far away, then there is a possibility that it will very quickly recombinate and high percentage, high probability is there. As a result, that will say form as a different character in the progeny. The student must be able to understand it by previously one thing we need to say, that is what is a wild type of characters and the mutant character in mainly in Drosophila we explained. Then with the help of a genetic map, they don't know actual genetic map. They don't understand that. What is a gene map? How they are though we are teaching in the Human Genome Project itself, still they will forget. We need to say that on a chromosome, we can draw a small diagram and we can say how that chromosomes are, genes are distributed on the chromosome. We can just explain even with the different colors of charts or different color papers. We can try to explain so that this independent assortment is also disproved by linkage. In linkage, there is no independent assortment. Only when recombination is there, then only independent assortment is proved. So student must be able to understand there is a possibility that genes can be inherited just together without being independently assorted. They will be segregated. So this characters kindly tell to children beforehand. So wild type and the mutant type. Then if we explain what is there in the book, that flow chart, student will take it as very easily. They will be able to understand. I believe this is what my experience. Teachers also can share your experience in this regard. How better do you find any other alternative options? You can share, teachers. Yes, ma'am. Jency ma'am wanted to say, I think.
Sir, I actually have not succeeded in this chapter, sir. That's why I asked.
Okay, okay. Now do you feel it's okay, ma'am? Now do you feel
No, when
comfortable?
I mention to the children, I don't
Okay.
feel I'm successful. In spite of using different colored chalk, giving explanation, I think I'm not effective all the time. I do not know why with regard to this
Okay.
chapter all the time. With regard to
Sure.
recombination, linkage, I do not know.
We'll make a group effort, ma'am. Together we'll all try to find a solution. We'll make all our group of children schools. We'll make the children to take it in a very easy way, ma'am.
Okay, sir. I actually thought I'll contact you afterwards, after this
Sure.
class. Whenever I'm going to handle the classes individually, I
Definitely,
definitely in trouble.
Sure. No. It's not like we're highly privileged to support, because, ma'am, yesterday I was saying we are all going to transfer this knowledge to the children. We are going to make them the sole beneficiary of all these things, all these efforts are the student, ma'am. I am also here for the students. You are also here for the student. We all want to take that in mind, ma'am. So it's not like you are troubling me, or it's definitely not like that. We need to support each other. Even when I have some need, I will definitely approach you. I will ask help from you, seek help from you. Yes,
Okay.
yes, yes. Amuda, ma'am. Yes, ma'am. How do you feel, ma'am? Any other possibilities you would follow?
No, sir. No, sir. I am in the class, so that only I did not on the mic, sir.
Okay,
Children
okay,
are
ma'am.
dressed for that, so that I am sitting here inside the class, sir.
Okay, ma'am. Okay.
Tomorrow they are going to write a physics now, sir.
Okay, ma'am. Oh, fine, sir. So this is that linkage. So it's really worked out for me when I explained, because in the beginning I don't go with the NCERT reader, teachers. The NCERT reader, of course, a very good book, but few things will not be there. They will assume that students have already understood, or it's known to the student, or what, we don't know. So few preliminary things are what is the in and around topics. First, if we explain to the children, And then when we enter into the core content, when I did that experimentally, when I did that, students are able to grasp that idea very easily compared to when I directly enter into the course content. They feel it very difficult to grasp. So we may all together will try to do in a different possible way also. We'll try to do.
Yes, sir. Even I
Yes,
too
teacher.
face difficulty in only in this particular topic. In the previous Mendelian inheritance, that monohybrid
Yes,
dihybrid will be
Hmm.
satisfied delivering. But this
Yes,
linkage and recombination,
yes,
even
ma'am.
diagram, we have to make the students to draw the diagram. Even how much we explain also,
Hmm.
right kind of explanation I could not able to give in their easiest way.
Yes, ma'am. Yes, I understand. Now we'll all together, even I will find some other ways also there. Let me share it on the group. Everybody will share it, ma'am. Okay. Thank you. You're absolutely right. That happens. Yes.
Yes, sir.
So this Morgan and also his next the Alfred Stuyvesant. Usually people ignore that person. The very big book also, it is given a very one page last line, the next page first line. That only, that is the maximum information will be there in the textbook, all our textbook. So we need to say that even NCERT, the question is there, who developed the genetic map, who mapped it. So we need to say that Alfred Stuyvesant, that concept little we need to highlight to the children, make them understand what is genetic map. Generally they don't have that knowledge. What is genetic map, how these genetic maps are done, though we are going to teach in human genome. That's what I already said. Whatever the concept should be taught before this principles of inheritance, that and all given in the next chapter, that is in the molecular biology, it is given. Now we cannot go to the human genome project while teaching this lesson. This lesson come first, then only we have molecular biology. So student has to wait till we complete genome project, then once again we have to tell, we need to recall the previous chapter and we need to make them understand. I have observed such a kind of difficulty in many areas in the reader. Many places we will find this situation. Then sex determination and all very easy. That chromosome map, they should know. They don't remember that. Chromosome 1 to 22, the autosome, and X and Y chromosome. Somehow we need to make them to clearly understand. Somehow we need to make them keep in mind this, how this flow chart of the mapping chromosome map is done. They don't usually refer all these things while they are writing about any genetic history or anything. They don't know the number of chromosome, what is the, which one is the biggest one, which is the smaller and all. They don't usually keep in their mind. But anyway, as we only know in a broader perspective, what are all the lessons are going to come in the future, what are all the needs will be there, we only will know. So we need to tell the children in the future it is going to play a role, very important role. Platoon, we need to put them in their mind. They don't remember even what are autosomes and allosomes. This that word itself they won't keep in mind. Generally in the classroom also, instead of saying chromosome number 1 to 22, XY chromosome and all, we can use those words itself. What is the technical word? Mostly we are not using that is true only. We need to say use that. We must use the word like allosomes and autosomes in a general way of communication itself. As I already have said, we need not to say that word T, capital T, small t. Instead of saying that, we have to say that dominant allele is there, dominant allele is here, recessive allele is there. When we say that, student will be that they will use the technical terms whenever they are writing, whenever they are in the regular communication also they will be using. Same way here also we have to use that autosomes and allosomes continuously, that we have to say. And we have to give the credit to Henking, how he has discovered that discovery also. Students are there are questions asked in the exams. Students need to be very stronger in that. When he was looking into the structure of the gametes, the male gametes, he has identified only 50% of the male gametes had a specific structure, whether the other 50% did not have that, did not receive that specific structure. So he gave the name, whatever the structure is there, X he has given, whatever is not there, he has nullified that, he gave that not. he gave that not symbol. So because of that only it has become x0. What students are misunderstanding, x0 they don't understand like xo, xy, they are confusing. They need little clarity in that. Regarding that, we will see one by one slide. You also can share your experience when whatever happens during teaching of this. You can also share teachers. That will be much better. We can discuss in a better way if it is known. So student must always say there are three mechanisms of sex determination. One is one organism will have both the chromosome, both the allosomes, and other organism will have only one pair of that allosome. Second one is one will have homozygous allosome and will be heterozygous allosome. The next one is again homozygous allosome, homozygous and heterozygous, but the gender will be reversed. So this idea student must understand. This chart also we can write it in a draw it on the board how they are actually determined. Here we can say that the mainly like what is heterogametic and homogametic means what. That word children again repetition only we need to put into the mind. It's a definition only, a simple definition. What is a male heterogametic and male homogametic, or female heterogametic or female homogametic? Both these words we need to say. First of all, there's a grasshopper is a very repeated example in the board exam. Many exams, if you can see, these questions are repeatedly asked. So we need to say exactly with the help of diagrams or somehow we can say that the males are heterogametic because they produce two different. They are responsible. Actually, heterogametic means they are responsible for the gender determination. That's what overall idea. Same way, homogametic, they produce the same type of allosomes, which they do not play a very crucial role in determination of the progeny sex. That the children must be able to relate. Same way in the human also, it's male heterogametic and female homogametic. We need to link with that how it is happening in Drosophila also. We need to explain. So children will be able to comprehend very easily. And third mechanism will be completely different, where male is homogametic and female is heterogametic. Examples of these birds. Most of the teachers would be thinking this and all very easy, because for teachers or the students, it's a very easy thing only. Nobody would be facing any difficulty in that. If at all anybody, please feel free to share teachers. But what is very important in this, if you see, the honeybee. Honeybee, 11th standard also they have studied. Do you can anyone recall where they have studied in 11th standard? Try to recall, teachers. In 11th standard also they have studied about this sex determination. A small information in the 11th standard itself. Can anyone try to quickly recall? In the cell division chapter, there is a word. Usually the cell division will happen in the diploid cells to make the haploid gametes, they say.
Yes, sir. Yes, sir.
There are some exemptions. In animals, male honeybee, in honeybee, in lower plants. In the lower plants, it is possible that mitosis will occur. Like that, there is one statement in the NCERT book itself. So teachers is really very useful if we teach them in the 11th standard itself that concept. Because they are studying nothing but cell division. There they will know more about the mitosis and meiosis very well. Yes. They know. When we are teaching there itself, if we put that idea to their mind, definitely they won't be facing any issues. Only one word, the parthenogenesis alone, we have to tell them when in the 11th standard. But that also is there in the 11th standard. The partheno copy is there. In the, what is that, in the auxins and all. Yes. Yes, there also we have mainly in this, growth hormone, plant growth regulators, there is a terminology. The auxins are very useful for stimulating this parthenocarpy in the plants, like that it is there. But we cannot explain the children all these things. But in 12th only, this parthenogenesis can be explained only in 12th standard. But 11th standard itself, if we explain this particular gametes, how this haploid and diploid gametes are there, how in queen bee, how it is happening, and in the drones, how it is happening. If we can say all this thing in 12th standard, they won't have any issues, because this topic, you can very well, you can see that only they accept the fact sex determination. All other concept in this particular topic is completely relevant to what they have studied in 11th standard only. There is no relevancy of this particular topic within this lesson or in any other lesson in the 12th standard. It is directly linked to 11th standard cell division only. So here itself, we need to put the mind children in 12th standard, 11th standard itself, how it is actually determined. Already repeated questions in the exam, that all the grandfather will not have sons, sons will not have father.
Grandson and that one. Grandfather.
grandson,
Yes,
yes. Yes. Can anyone say, teachers, what is that? How can we explain to the children?
Because only without undergoing fertilization they reproduce, no, sir. So
Yes.
there is no father, directly grandfather and the grandson is there.
Yes. So we can add on to that, ma'am. Very nice, ma'am. Yes, Samudha, ma'am. Thank you. So we can add on to that, that the male honeybee has born to them a mother who has the father. From the father only she has born. From that mother, this baby has born, the male honeybee has born. So the male honeybee has the maternal grandfather is there, but there is no father, so there is no paternity itself is not there. We need to put them to the children. The paternity and maternity, that word we need to make the children to understand. The male honeybee was born to a mother who was produced by a male honeybee, the grandfather. So the all male honeybee has a grandfather. Same way, the male honeybee is not going to involve in fertilization. It is going to use a female honeybee with fertilization, and that female is going to give birth to a male honeybee. So it has a grandson. So we need to make the children understand that pattern. Even sometimes I use Tamil also, that grandmother from mother side and father side, that and all will make the children to understand, means they will like it very much. They will be able to easily grasp that concept of this haplodiploid sex determination. Very important concept in the 12th standard. Yes, teachers, anything you want to say? Is it okay for you? Do you have the challenges anytime? Did you have it? Gayatri ma'am,
Sir,
same
no,
ma'am.
no. Yes, sir.
No.
No,
Now
no.
is it okay, ma'am?
Yes, sir.
Thank you.
Yes.
Yes, thank you. Thank you. So mutation, next, very important. So before mutation, we need to tell to the children, not mutation is not always lethal. There are lethal mutations are there, that is different. Generally, mutations will occur throughout the organism. That is the reason why this enormous biodiversity is created now. Even within a human population, we are all not looking similar. We are all different. For that, there are two contributions. One is sexual reproduction, where the segregation happened. Second, most important contribution comes from mutation also. Because of the mutation only, today we have lot of diversity. Within the genetic diversity also is more in us. That is also because of this mutation. Student must understand. Sometimes it can lead to lethal mutation. That will lead to various negative impact on that organism. But otherwise, the mutation is here. Example of the sickle cell anemia. Here itself to teach the children. Here only mainly it is there. That is the what is the changes happens in the point mutation. There are two type of mutation, like point mutation, frame shift mutation, and all we can tell to the children. The example of this sickle cell anemia are very important questions asked in the repeatedly in the board exam. That is, what changes happens in the DNA level and how it affects the RNA, and because of that, what changes happens in the amino acid coded. As I already have said, again another question, the student will need the codons concept. Without knowing the codon, how come they will be able to say what is that mRNA, the DNA and all, teachers? Am I right only, no? Am I saying the correct thing?
Yes, sir. Yes, sir. Correct, sir.
Okay. So they don't know. They next chapter only we are going to teach them all this codon concept. But here itself we have to explain them that the sixth position, what happens, and all these things we have to explain. Generally, I will hold this topic, teachers. What I did, just I'm telling, sharing with you. I will hold this topic once when I am teaching that the molecular biology. Later, I will come and complete that because that will help the children to understand in a more easy way compared to how I am now. If I teach during this, when this first time they are learning, I don't think they will be able to understand. So usually I do that. It's all your decision what you can do. You can say that. So what happens to the C, the thymine here also? Let us not say that C is replaced, T is replaced and all. We'll correctly say this: thymine is replaced with cytosine, or cytosine is replaced with thymine. So that word we will use. Put it in practice that they must be addressing the nucleotide only. They should not merely addressing the letters. They should not think about the letters. They must understand about the nucleotide. So repeatedly, we need to put that into their mind. And here, whatever if you teach, they won't understand why. They know adenine, thymine, and all, they will know. In the mRNA level, when we say that uracil has to be there, they cannot understand. Uracil, they won't understand at all why it has to be changed. Though they have studied in 11th, a little concept they have studied, they won't understand. Only after molecular biology, we need to come back once again, and we have to make the children to understand, which I am following. It's, you can take that. But frame shift mutation, children will be able to understand easily by giving an example of the base pair. We can say, even we can put a words of CBSE, there is no mandatory of this particular nucleotide sequence, no need to be written here. With the even letter, in the book itself, there is a Rom has cap, Rom has red cap, like that, and all. What is there on the screen also, you can see that beast, feast, breast, best, beats, like that, how the different types of frame shift mutation. One is a normal gene, beast. It can be substituted by some other nucleotide, or some insertion can happen, some deletion can happen, or the inversion can happen. Like in a different way, the mutation can happen. That this example also can be quoted to the children. So how different same letter, when we are changing certain things, how the meaning changes. Same way, when a nucleotide sequence, if some changes happens, it will lead to the changes in the amino acid, which will definitely code for a different protein that we can tell to the children. This also another example. And I think Amuda ma'am only has sent one very nice that mnemonics for some concept. You only send, no ma'am, Amuda ma'am?
Yes, sir.
For transcription you have sent, I think. Am I right? Mnemonics.
Yes, yes, yes.
Yes, it was very interesting. I'll share with all the teachers later. Yes. So like that, we can have some other examples we can give, or textbook examples also we can give. Next one is, what are mutagens? This the students will ignore mostly. We also will not give equal weightage, but we need to understand, make the children understand. Mutation cannot happen all of a sudden on its own. There are some factors which is essential for the mutation to occur, make it happen, like physical mutagens or chemical mutagens, something the children must be able to understand. And mustard gas and all has caused a lot of, during World War. The children, we need to explain the children, during World War I, the mustard gas was used. And mustard gas was used for killing the enemies, making the problem with the enemies, controlling the enemies population. Do you know which scientist has given which animal? One animal only protected the people from mustard gas. Do you know which animal, teachers? Do you remember? Actually, what will happen if any other gas is there? The people will come to know immediately. Then they will protect themselves from that. When mustard gas is released in the air, we won't come to know immediately. It will take some time. Many hours it will take time for the mustard gas to show the reaction in our body. By the time already we would have received the mustard gas, it will have a lethal effect on the body. To avoid to know the presence of mustard gas, a very important animal has helped. Can anyone recall, teachers, which animal?
Rat.
It is okay. It was actually snail, ma'am. snail will be able to find the mustard gas. Yes, you need to believe the snail only helped and saved many soldiers from the mustard gas attack during the World War. So this and all, if we tell to children, they will feel more interested about how that our biology system has worked throughout the history. They will be able to understand.
Even for deletion, minimator disease, that also
Yes,
can be.
ma'am. Yes,
Minimator.
exactly, ma'am. Yes, exactly. And
Thalidomide.
yes, ma'am. Very nice, ma'am. Very nice. Yes, yes. And also we can take the help from this previous, in the lessons of cell division and cell cycle. What are all the examples are there? That also we can take example here and we can quote it for the children's sake. So that's all so far. Whatever we have seen, mainly the repeated questions, important questions, we have mostly covered it, teacher. Next, we are going to see that another very important one, pedigree analysis. Students here will not feel difficulty and all. Students will only feel they have to keep in the mind of that particular the symbols, various symbols they must be able to keep in mind, and it is very important for the practical point of view. Did you all note, teachers, what changes in the 12th standard practical? Do you remember?
Yes,
Did you
sir.
notice?
That soil, water source, that practical work they have deleted.
Yes, ma'am. Which very easy we have followed for the past many years has been
Sir, but why
removed.
they have given this quadrat method, and how do we take the students to the field in case if it comes in minor experiment?
No, ma'am. Actually, we are ignoring that plus experiment, isn't it, ma'am? So CBSE knows that for past many years, it is observing that we are ignoring it. Actually, no need of field and all, ma'am. You can directly do it in the laboratory itself using a tray. In the tray also you can grow. Usually, no need to do in the field and all. Using a tray, we can make a smaller scale.
Yes,
When
yes.
time permits, we'll discuss regarding that. Also, not an issue. Even we can go with the chart, a small chart we can make. From the chart, the student must be able to identify. There is nothing but they have to identify, count. They have to apply the calculation formula, and they have to find the answer. So it's very easy.
Analogous, homologous organ and analogous organs are included, no, sir, this year.
Yes, ma'am. That has been there for many years. Only for COVID-19 it was removed. But they have removed completely this, soil experiments
Soil
and
and-
water experiment. They have removed. That is really a shocking one. Why they did
Can
like
you
that?
share that one, sir? How to do inside laboratory? That
Yes,
one
yes,
can
ma'am.
share,
Yes,
sir.
yes, definitely. We are all doing that. Definitely, I'll share it with you. Even if possible, I'll take a video and I will share it in that group. No issue.
And they have included also this homology and analogous
Yes, ma'am.
organs.
But that is not actually included, ma'am. It was there for last 2-3 years before itself. It was there, but that is only choice. We need. There are 11 spotters, I believe. We are going to only take 7. We can maybe we can ignore also if possible if the external is agreeing with that.
But last year we didn't do that, sir. We didn't.
Yes, ma'am. We didn't. Last year many are deleted, no, ma'am. Last year
yes.
many deleted. Yes. Even the salivary amylase also was removed only.
Yes, sir. Yes.
Yes. Okay, ma'am. So next problem will be with the pedigree analysis, where only the simple memorizing. The other things and all very easy for the children. They first of all they won't remember which symbol for male, which symbol for female itself. They won't correctly understand. And here, what will be the issue? Directly with this flowchart, there won't be any need, ma'am. But when they are explaining about the autosomal linked or sex linked, there the students will be having issue. There they will be the students will be facing a problem how to deal such a kind of question based on pedigree analysis. There it will be an issue. If and all you want, tomorrow I will make that particular one for you. Whether genetic disorder you want me, then I will add it tomorrow. What do you say, teachers? Do you want it?
Genetic disorder means next topic, sir.
Yes, ma'am. I didn't include today. I thought based on your decision, I will include. I thought if you feel you want, then tomorrow I will include that.
Yes, sir. We can include.
Sir, tomorrow discuss the exam point of view questions, sir.
Yes, ma'am.
Yeah.
Definitely, ma'am.
13th and because we can revise for children now, sir. In the
Okay, ma'am. This
13th
term 2
and the
syllabus.
15th chapter, 13th and the 15th.
13th and 15th, ma'am. Okay, ma'am. It's a very, you made my job
Because
very
after
easy, ma'am.
Monday,
Sir,
after
actually,
this,
tomorrow a little bit we can go with the theory and then more we can concentrate discussion parts, sir. Like that 6 lessons also.
Okay,
What are
ma'am.
the
Done.
important points? Yes, sir.
Are you all handling 12th standard teachers? Because others should not feel like they are opted out. That's why all the teachers handling 12th standard, Jansi ma'am and one more ma'am, all are
Yes,
handling
sir.
12th standard, right? Yes,
Sir,
yes. Then no issue. Sure. Okay. Then tomorrow we can go with the 13th, 15th is a very the organism and population. Then this lesson and all the chapters also will discuss, and evolution whatever is possible we will try to. Tomorrow we'll discuss the teachers. Now next one is the evolution chapter. This year it's going to be a challenging. Believe me, your experience how it will be. Even for my experience, I'm going to share that students are going to be exhausted by the time we complete molecular biology. All the 3 chapters in a queue, all are vast chapters. All the 3 are very vast chapters. When we start in the month of June 15th or something, I will be able to start, I say, and it will complete only in the month of maybe in the August first week or sometime. I don't know how many days it is going to take. The present batch, that issue will be there because ma'am, 10th standard they don't have evolution topic. Am I right, ma'am?
Yes, sir. Yes, sir.
10th,
It was there, but it was deleted actually. The
yes,
topic
it was deleted. Yes, that's what. And for board exam, it was deleted. As a result, students are going to have a very fresh mind. And moreover, this is a history. So the history will be, as we all know, how the history will be for the children. So we need to try our level best. Even I will be sharing whatever my experience is there. I will be sharing. Together we'll try to bring a better understanding of the students. So we can start this lesson with, instead of calling the origin of life directly, if we enter into history, is historical perspective, I believe students will be able to student will become little boredom in the beginning itself. The more, instead of we motivate the children, it will become like a demotivating for them sometime. So we can show the children. There is a very one nice video teachers. Maybe some of you would have seen that.
Sir,
It's
actually,
about
this lesson lecture mode itself will not work out, sir. We have
definitely,
to show
ma'am.
some videos, PPT only. Then only the students
Yes.
will understand. Lecture mode, if you take chalk and if you write on the board, surely they will not show some
Yes,
videos.
exactly, ma'am. Yes, same experience. For almost everybody will agree with that. Almost same experience only.
Yes,
But
sir.
teachers, I have seen one video. I have shared with the student also. Now I couldn't able to start it now. For past 2 years, we are not teaching this. That's why I don't have it now. That cosmic theory. There is one lesson. This is called cosmic theory, a space-time odyssey. You can search it in the Google also. A cosmic theory, a space time odyssey. Like that one. Video is there. It's a very Discovery Channel, National Geographic video only. It is not any local video, so no need of any the information and all will be very accurate. So no need to worry about it. So you can download it. It goes for more than half an hour. So we can try to make it as smaller as possible. It will give the children a very interesting idea about. We are very good in discovering what is there in a very far universe. In the universe, what is there in a very far away places, that we are able to identify, discover, but we are not able to discover about our own self, whether we have understood about our own self or the living organism present on this planet Earth. It's a very big question mark. So in that video, actually it is explained, very nicely explained, that we are all a very small point in the, when we compare in the universe, we are all a very small point. They also make a calendar, they make a very calendar in which the life of the earth is started on the 1st January night, that is 12/1 the earth was formed, the planet, the Big Bang theory has happened. When the human population was formed, if you see, after that 1 year, December month, or 31st December night 11:59, only human was formed. That is after that nearly 1 year. The 1 year is going to end within 1 minute. Very short, very few years before, when we compare with that universe calendar, our human population was formed just 1 minute before only. Like that, it is a very nice comparison. Maybe when I explain it, maybe not so that much effective, That video has to be seen and it has to be appreciated. I will also just find it in my old system if I can retrieve it. But sharing will be a problem. Why? Because it's a 30 minutes, more than 30 minutes, it will be there. Please try to watch that and show that to the student, and student will definitely appreciate the learning of evolution. They will definitely appreciate. Practically, it really worked out for me 3 years back, I believe, before that corona batch, until before the corona batch. After that, we couldn't use it. The space time odyssey, the name of the topic, this is cosmic theory, a space time odyssey. It's a National Geographic channel only. They only actually made this video. Yes, teacher, shall we go to the next one?
Yes, sir.
You can try to find it, and you also let me know. Okay, next one, the origin of life. First, what happens this lesson, they try to find how that life has originated on this planet, how this planet happened, Big Bang theory, how this universe was originated, how a single huge explosion has happened. All this thing, when we say tell the children, they will mostly they will be passive only, I believe. They won't be able to comprehend all this thing. What is that Big Bang theory itself? It is not understood by the children clearly. So we this and all not so important. Also, we can just give them idea what is this and all happen. But very importantly, that origin of life by this chemical theory of evolution. All the other things are the theory of special creation, cosmozoic theory, panspermia, like different things are there. They are all not a proven methods. They are all just a philosophy and the experimentation. Student must be able to understand, and student must clearly understand, studying about origin of life cannot be a experimented thing. It can only be, in most of the cases, it can be only a philosophical perspective only, the hypothesis only. There is no possibility of experimenting many things. Big Bang theory also was tried to be experimented, but it was utter failure. Can anyone recall, ma'am, the Big Bang theory happened, experimented?
1931.
No, ma'am, recently, lot of this reaction from the people of the world, especially from India and all. What happened? They said something, the earth is going to be destroyed because of the Big Bang theory. Do you remember in Sweden it was done? They have made tunnels under the ground for few kilometers. They made this electrons to bombard. Very quite recently it was happened. Even it has caused a lot of riot from the people. They didn't agree with that experiments. That and all we can tell to children, how the people are, even in 21st century, people are still superstitious only. Those things we can tell to the children how it is happening. Then very important one is this chemical origin of life is the one which is mostly accepted. Otherwise, all the other things like abiogenesis, biogenesis, panspermia, special creation, all these things are ruled out, because they don't form any practical or any practical evidence for to prove that. They don't have that. So all these things were ruled out. Only at last chemical theory of evolution is accepted. Here we need to explain how it was disproved. Say for example, abiogenesis. When there is nothing is there, how come spontaneously something can be created? That was disproved by Louis Pasteur. Are we all explaining the children about the pasteurization, teachers?
Yes,
Yes,
sir.
sir. I'll
Yes,
explain.
please,
Sir.
please. Very nice, ma'am. Yes, please.
I'll tell
This
about,
person
if they have chance, you have to visit Pasteur Institute, which is opposite to Ooty Botanical Garden.
Microbiology, father of microbiology.
Yes, exactly, ma'am. Yes, many information you are sharing. Very happy to know that. Yes. So we need to explain. And he is using his, that a special apparatus he has used, an open flask with, the neck closed. It can be closed. And he has tried to prove that this particular abiogenesis theory is definitely not possible. Then biogenesis. Biogenesis is somewhat accepted. Biogenesis is from the life only, the life develops or forms. From one life, the other life is formed.
He found some maggots.
Yes.
Francisco Reddy. He
Yes,
did that
ex
answer.
exactly, ma'am. Yes, yes. Francisco Reddy, Spalzani, and Louis Pasteur, all these people together, they tried. But when they were keep on holding the thread and going, the question again comes into my mind: how was the first organism on the planet was maybe formed? Of course, the biogenesis they are able to explain at the present scenario. From one organism, the other organism is formed. We can even go with the Darwin's theory of evolution. Everything is okay,
Yes,
but they are not able to answer how was the first life originated on the planet. None of them were able to actually explain. Then many philosophers they said that the spores came from planets that fell on the earth, and because of that actually the life has originated, or the special creation. Of course we are all god, we have trust on the super, some things are superior. But when it comes to the science point of view, we cannot just believe will not work in science. We need some as solid evidence for the creation. When we have that solid evidence, then we can agree with that. Anything supernatural power or anything, we can, in that science also we can believe. Next one is chemical evolution, is a very excellent and very perfect by Haldane and this Oparin. This theory is the one most accepted by many scientists, and that's why the chemical theory of evolution can be a particular a theory that can support the life origination on this planet. Again, here also abiogenesis happened, then only biogenesis happened. So abiogenesis, simple inorganic molecules and organic molecules, life has originated. From that particular life has evolved into many stages, and today whatever the life biodiversity we are seeing, they're all the outcome of that particular theory. And this chemical theory of evolution was also experimented by Miller and Urey. Please, in 10th standard itself, kindly teach this, teachers. 10th itself, there is a concept. Of course, this year we will be having. So 10th itself, that experiment, it is there in the book. Have you noticed, teacher, that
Yes, sir.
experiment is there in the book?
Yes, sir.
Yes.
Very
Please
important.
kindly, since we are yes 11th to 12th we are handling, kindly in 10th itself little knowledge put them in their mind, so that they will know when they come to 11th 12th standard they won't have much difficulty in understanding how, what was the setup, what was the experimental setup, what are the things needed in that, how different hydrogen water vapors and all heated at 800 degrees Celsius, how amino acids are formed. Definitely amino acids are formed. These amino acids are there later packed and they form protein. Then from the protein everything has happened. So these are all is a proof. Experimentally also it is proved that chemical theory of evolution can be the most appropriate theory of evolution. Now next one is how this cellular life has originated later. If we see that, we don't know, but we have some approximate. Children must understand everything is approximate values only. Non-cellular life was there for around 3 billion years ago, it was there. Then how this metabolic capsule, as I already have said, some these molecules were covered, then inside that it become a macromolecule, and then it has become a life. So how this process happened, the children must be able to understand. So if you are keep on talking about evolution, there is no answer will not be there. So we need to say evidences are needed in science. There only comes the evidence for evolution, starting with all this paleontological evidence, morphological and anatomical adaptations, adaptative radiations, biochemical evidence, embryological evidence, and mainly natural selection. So each and every concept together, one question we can expect in the board exam from this particular topic. Mostly they ask about this, analogy and homology, mainly they ask. And also they ask about this, the experimentally, how that is happening. Mostly that question is mainly asked in the exam. Regarding this also, one by one now we'll see. Teachers, you yourself, your suggestion for assignment, what shall we do today? Something useful. It's not like for the namesake, I also don't want to give. You also, your waste time no need to be wasted for that. Something very useful, what shall we do?
So time scale of year.
Very nice. Exactly. Can we teach us the universal, it is in Google also it is available. We need to compare the universe calendar with the evolution. You will find very well, you will find a 12 months calendar and it will be there. Can we all do the teachers if you are agreeing with that?
Yes, sir.
Jansi ma'am?
Yes, sir.
Yes, please, every teacher, please, it will be very useful for you when you are teaching this lesson in the 12th standard. Not too much. Now in the month of July, we will all be teaching this lesson, I believe. At the time, that calendar will be very much useful. Let us make that. The assignment will be like, now we need to make that universal calendar. Only 12 months will be there. It will be very clearly mentioned. The planet was originated on January 1st at the 12/1 of that particular year. After passing all the 12 months, at the last month, December, last date, December 31st, night 12 o'clock, before 12 o'clock, that is 11:59, human has evolved on this planet Earth. A very clear depiction will be there. Please try to do that. I will just note it down. And also the video I will try to find if it is available, the link or how to find that I will try to share it with you. So next one, this paleontology. Please kindly give the career guidance also here and all. When we are teaching them completely, they are blindfolded. The only job they know after becoming a biology student is doctor, NEET. They close their eyes like a small children. They will be repeating the same thing, doctor, doctor, doctor. Kindly please give that idea to the children that these are all a very wonderful job with very less competition. More promising jobs are there. Not in India, also abroad and all. There are a lot of opportunities are there. Please put that into the mind of children. This paleontology, what is that? How it is different from this? That what is a fossil? How it is different from the archaeological studies? How they are different? Archaeological studies is like artifact, finding the different equipments or materials used by the people, not actually about the organism. But paleontology is about the organism, finding the organism lived in the past, and we need
Sir,
to tell the children.
we are having one great place, sir, to identify many. Daily one thing they are identifying, sir.
Yes,
Agalvara,
ma'am.
that is
Yes,
Kizhadi.
Kizhadi, yes, ma'am. Kizhadi, yes. Kizhadi is actually archaeological site. So children must be able to understand the difference between these two.
Paleontology.
Yes, ma'am. They must be able to understand. So how these fossils are formed, they don't understand. Mostly they think like, organism will be just there. So they need to understand that how the imprints are there. How that imprints are when they stuck in between the stones, how they are making. Actually, we did that for as a science exhibition. We made the children to make the fossils of different animals using the stones on the sandstone. They made it actually. We can try that also to the children if they make, and they can keep. We can keep it in the laboratory. We can use it again and again, repeatedly, year after year. We can use it, teachers. Then how these evidences are there? How the paleogeny evidences are there? How they are helping? Say for example, when we find the woolly mammoth, how we are relating it with an Asian elephant and an African elephant. Like that, we can make the children to correlate the animals that lived in the past. When we find the remnants of those animals, we are able to link their relative in the present time. Who is which animal is relative, that we are able to find it. And based on that, we are trying to find the evolutionary history between different animals, how they have evolved. Here, kindly give the example of Archaeopteryx. That's a very important example. Yes, teachers. Are you this bird, any specialty can anyone say? This particular organism.
Connecting links are between reptiles and birds.
Very nice, ma'am. Yes. What is that, ma'am? Any Gayatri, ma'am, also can anyone can say? What are the features were there in that particular animal feature, that organism?
Archaeopteryx, sir.
Yes, ma'am. They had a tooth in their, we need to ask the children the question. They had the tooth in their beak. Is it the character of reptiles or not? We need to ask the children. They what is that answer? We know that it is not a character. The reptiles should have teeth or not, teachers? Mainly the teeth, presence of teeth is actually reptiles. Amphibians, the birds will not have it. Then same way, the wing is not used by this particular animal for a feather protection from the wind. So we need to put such a kind of things, and we need to make the children understand. So today with this, I would like to wind off, teachers.